This has been an incredible time. Steve has pointed out that basically I'm a Canadian hewer of wood and drawer of water, and I'm going to talk about generating data for people to work on. Okay, so one of the things just to start, and it's a great advertisement. This is our new Institute for Personalized Cancer Therapy. We've just capped the top floor. This will be open, and the goal here really is to change the way in which we manage patients. You heard from the previous speakers some of the steps along the way. I'm going to tell you some of the things we're doing. I work with industry. Now, these are not where I have funds from, but I put this up for a reason. One is that I need it for CMA talks. But more importantly, we've been hearing about academics collaborating broadly with each other and sharing data. If we are going to help our patients, we have to change the way in which we work with industry. So we go back much more freely, greater data sharing, greater information sharing. And so this is something we have to figure out how to do while maintaining an appropriate conflict of interest coverage. So I think you've heard about this, and I just use it to talk about one little point that I don't think has been discussed today. We're moving forward to targeted therapy for cancer, and the ability to characterize what is going on in the genome of a patient has improved at a level and a rate that I think is just almost incomprehensible to most of us. But the opportunity to do and know what's going on has changed. But the question that I think we're not asking often enough is, so if we target those events, why is it that they will have a greater effect on the tumor cell than on the fact that all of the pathways, all of the functional events that we are targeting play a role in normal biology and are important most of the time? So what is it that generates these vulnerabilities and the Achilles heel of cancer? Can we begin to understand that in a way that we haven't done in the past and move forward? And we've heard some hints, I think, about this earlier. Now, I'm going to start with our clinical genomics program and then end with how we're building on top of this. And our goal in the Institute is that all MD Anderson cancer patients with no standard of care that is curative. Now, to me, this is a little bit of a frightening statement. That's 30,000 new patients a year that we need to figure out how to manage. Right now, and in the last, I guess, 3 months, we've put 2,000 patients through the complete program that I'm going to show you today and are starting to get data back as to where this is going. But scaling that to 30,000 a year is an engineering challenge that is going to be a major problem and important. And to answer the other question, this is all grants and philanthropy, no funding coming back and saying that this is yet ready to be billed for. We now look at, in our initial screen, for actionable targets, any aberration with a targeted drug in the clinical trial or late-stage development, any predictor of outcome, anything where there's something coming down the pipeline. We characterize these to exquisite depth. We have chosen in our patients to do ultra-deep sequencing as our first step to capture heterogeneity. But this gives us an idea of where to go. Turns out that less than half of our patients sit on this side, have a targetable aberration present, something that we have even a vague hint of what we could do about it. And this really does sit with what we heard from Eric. We're going to need a better information management system to find out what to do. Some of those patients go on to standard of care, a mutation in a gene with a drug that we've shown that work fits, amplification of HER2 and a HER2 drug. The biggest excitement is we are filling clinical trial cohorts in an incredibly rapid and efficient rate. And further, we're able to do some of the things that are truly challenging. There are 4 different companies with drugs out there targeting a molecule I'll talk about throughout this talk called AKT. AKT mutations occur in less than 1% of patients. You could never match a trial unless you did this up front. Test thousands of patients. And even if it's 1%, I'm going to find enough patients to test the hypothesis that an AKT drug and an AKT mutation will work together. That multiplexing done without a specific trial in mind, an umbrella approach, is going to be necessary to make this truly efficient. And then we heard from Eric about N of 1 trials, the concept that each patient could be directed to their own therapy, even if we have limited data to support that, if there's no other alternatives, with data capture at the end, not to say does that drug work in this patient, but rather does the process work and should it be promulgated for more patients. But I do want to talk about this side a lot more. What are we doing when we don't see an aberration? We go much more deep in sequencing, and I want to show you about our high throughput biological validation approach aimed at figuring out what to do for these patients, some of the excitement and some of the challenges that are perhaps frightening. Now, I'm going to use the PI3 kinase network as my example throughout this talk. This is a signaling network that links the cell membrane through a number of signaling molecules down to most of the important functional events in the cell. I want to focus on a couple of these just at the minute because I'm going to come back. I just mentioned AKT as a key node and a key target. The catalytic regulator of the pathway is P110 here. The adapter molecule, P85, regulates P110, and the major negative regulator is called P10. And I apologize, P110 and P10 can sound a lot alike, so I need to be careful. Now, I will point out that part of this story is going to be that the textbooks tell you from all of the studies in the world that P85 is an adapter and regulator of P110. Since it's in the textbooks, it's clearly wrong, and this is part of the story, is the fact that a lot of what we think we know, and I go back to our first talk today, is wrong. It was contextual, and I'll come back to this several times. Now, nature has told us that this is an important pathway to go after. There are more mutations in more targets in this pathway that activate the pathway than any other known pathway. And I say activate because it's much easier to target a pathway that is activated than one that is inactivated. And further, it's chock full of kinases that we know how to target. Industry has figured that out. These are the drugs that are currently in clinical trial. I think this slide's about a week old now. There's 2 that have fallen off, 3 that have come on to the top. And I've drawn the pathway a little differently. We have the growth factor signaling arm and then the energy stress sensing arm. We've heard a lot about bioenergetics. This is the key arm that detects what's going on in the cell bioenergetics. Lots of interesting drugs starting to come out on this side. They happen to be some of my favorite drugs. Down here at the bottom, resveratrol, ECGC, and xanthohumol. Resveratrol is the cancer preventative that's in red wine. This is the one in green tea, and this is the one in dark beer. So as long as you drink dark wine, dark beer, or dark tea, you're in good shape. And I really helped my health, moved myself off of one of those peaks to another last night because we had some great red wine. Does this work? And so this is just one piece of evidence that says this process could work and some of the challenges that I want to deal with today. We took 1,000 patients and tested them, and all patients that had a PIK3CA mutation were offered in our phase 1 program, where we had no other alternatives, targeted therapy against that pathway. And there were many combinations here, but the overall combination was a targeted therapy in the pathway. And what I want to note is in this waterfall plot, patients progressed, some did well. In an early phase 1 trial, we expect about 10% of patients to have a RECIST criteria change in size of their disease. This was 30 to 40%, so it works. The idea of linking the two together has the potential to help. We need more, but there's a bigger message here that I want you to pay attention to because I will come back to it several times. There's a color coding here by disease. Turns out if you have a PIK3CA mutation in bowel cancer, you're not going to benefit from the targeted therapy that was used. If you happen to have a PIK3CA mutation in this group of cancers, which happen to be women-specific in this set, cervix, endometrium, breast, ovary, you will respond. So there's a difference related to the intrinsic gene expression pattern of the lineage. There's also the question of the combination of mutations. We talk about one. Every tumor has 10 to 20 different mutations that matter. How are we going to integrate that data? And we're having a discussion with a pathologist earlier. The human eye has been integrating that mutational landscape for years by looking at what happens phenotypically because there are only a limited number of phenotypic changes that reflect all of those early events. But the other part that we see here is co-mutations. We predicted that RAS or BRAF mutations would be a resistance marker for PI3 kinase targeted drugs, and we had a great hypothesis, and in bowel cancer that's the case, and then we looked at ovarian cancer and our hypothesis was completely wrong. The data says go back to the drawing board and rethink. So, sounds great, we're moving along, but this whole area has had an incredible amount of excitement. The promise of personalized care is incredible, and we've made great steps. However, for almost all of the patients that we treat, even if they have a biomarker that says they should respond, less than half of the patients respond. Secondly, the benefit is usually short-term. One of the biggest excitements, new targeted drug in melanoma, the Plexicon inhibitor. 80% of patients respond, and after 7 months, every single patient has recurred. Not one patient, as far as we can tell, has a durable response. So we've got to understand what's going on. I'm not going to talk about the resistance mechanisms today, but I'm going to pay a lot of attention to this concept of rational combination therapy. And we heard about this earlier. I normally attribute it to Yarden, but I'm going to change this. And this is really the concept that systems are robust to single perturbations. I've had enough caffeine today that a number of my cells had to go quiescent, so they didn't get killed by that stress of turning on the PKA pathway, and they could do that. But the expectation is that tumors have decreased robustness because of the number of genomic aberrations. And if we start to hit multiple pathways in a rational manner, where we'll get these interacting together, we may have that therapeutic index that is critical. So, this is my systems biology and why we need it. I started in this a long time ago, and this is how we used to draw signaling pathways. There was an input, there was a black box, and there was an output. Now that black box has had king calcium in it, PKC, PI3 kinase, whatever it happened to be I was working on at the time was the black box. Well, everybody was excited, and this is a simpler diagram than some of the ones we've seen, when we could finally go from a receptor tyrosine kinase down to the nucleus and say, yep, we've figured it out. Now we have this sense, an input and an output, and a lot of things happen in the middle, and it is simply too complicated for anybody to understand. It simply gets integrated and then something happens. And when we talk about a normal cell, it's tough enough. If you have hundreds of mutations in a cancer cell, it may be ununderstandable on a single cell basis. We have entered a case of information overload. Our high throughput technology, I think most of us would love to say we're learning from it. It's data. It's not knowledge. It's not information. It's simply data. There are too many connections and too much information to integrate and visualize. The bigger problem, much of this data is contextual. I don't believe scientists faked it, but rather it was in the one little system where it was identified, and that probably has little relevance to reality, and it's all static. There is so little time-dependent, spatial, homeostatic loops, pathwayness, cell-cell interaction data that we're in real trouble. And finally, cancer is a real mess because the pathways are rewired. And what we learn in normal cells, what we learn in Drosophila, is going to be hard to link to animals. And so I think we're right back where we started. We have an input, we have a black box, and something happens. Okay, so the goal here really is to start to apply the concepts of systems biology to that challenge, to link the mathematical sciences, the biological sciences through a bridge, and really two different goals. One is to build these robust predictive mathematical models that can allow us to deal with the amount of data, and also to help make sense of it and make predictions. I also agree with Steve, systems biology is a data-driven process. And my main role in life is to generate data that hopefully other people can make sense out of. I think he was trying to imply I wasn't able to do that myself and that I needed the modeling, but that's okay. So this is one of the things that we've started to do with the PI3 kinase pathway. Now this is actually a differential equation driven model, and I won't show you the equations or their solutions. And this actually is a pretty big differential equation driven model because of the complexity and it's data demanding. But there's some neat things that start to come out of this in terms of targeting this pathway. The first is that there are homeostatic loops that if I target one molecule in the pathway is simply going to laugh at me and find a way around it. There are unexpected components. We cannot explain what we actually measure experimentally from what we think we know. So clearly I woke up and found out I was wrong. But some of the other things are important. Sensitivity analysis allows you to predict the consequences of perturbing a node in that network. PI3 kinase and AKT, where most of those drugs we're targeting, are not rate limiting. Yes, if you mutate them, they're oncogenes. That does not mean they are rate limiting, and that is a fallacy that we have to get over. That means we have to knock these down in terms of a therapeutic approach at least 90%, and most of our predictions and our real data now say 92, 93%. Our current drugs can't do that. Once you get anywhere near that, the toxicity is rate limiting and we're in deep trouble. So we've got to rethink how we do that. And the other is we now know that when you perturb this network with the drugs, there are mechanisms that are reduced that bypass the network. In a whole other talk, I could come back and talk about how we have got a high throughput program to identify and target those bypass mechanisms. But I want to do a little different story today. By the way, all of my data slides will have the people who did the work. I don't do any of this. I bring the money in to let them do it. I do the talks so we can get more money, and I write the papers. Okay. So we've talked about big data and big data efforts, and I think the biggest data effort right now in the area of cancer is the Cancer Genome Atlas Research Network. People who really are sharing data efficiently. All of it is in multiple places where you can bring it down. And we're generating the data for the crowd to work on. Now what's been going on in the first part of this was a focus on DNA and RNA. And over the last 3 years now, we've added a functional proteomics arm to this with a technology that is able to deal with the scope of the TCGA, 20,000 samples that we have to characterize from very small amounts of material. Now, why did we want to look at protein? Well, it's fun to look at DNA and RNA. It's easy to do it. However, if we look at DNA copy number and mutation, it's only about 60% of the time that is reflected at the RNA level. If we look at RNA levels and protein levels, on a good day it's 60%. I only put that up there because people yell at me. We've actually done the analysis, it's lower. But if we want to start talking about protein function and impute that from what's happening at changes in the DNA level, we really have limited power today. So we wanted to put protein into the network. We decided many years ago to use the reverse phase protein array platform. There are other platforms. This one, however, allows you to deal with large number of samples and small amounts of material better than any other platform. And it's best thought of as a quantitative, high throughput, multiplexed, inexpensive ELISA assay. Lots of things have to happen to make it work, but basically we put here 1,000 breast cancer samples, 1,000 controls, on a single slide and measure what happens for one analyte. This happens to be a PI3 kinase pathway member. We developed that, and this is just one experiment showing you the linear regression across different days, different samples. It really is incredibly good. By the way, that is 1/16 of one cell spotted on the array here, saying we have that degree of accuracy. And we've started to build, as you heard from Steve, the Cancer Proteomics Atlas. We've run over 3,000 breast cancers, 1,500 leukemias. We really are beginning to get the information trove that will allow us to move ahead. We've also run over 500 cell lines. We're running the Cancer Cell Line Encyclopedia that you've heard about to start to allow us to link what happens in cell lines to patient samples. And indeed, we can now take any patient and predict what cell line in the world at a proteomic level is most close to that patient. We've run over 70,000 samples in total. That data is, in general, freely available. That's not an offer, it's a threat. So here is our study from the TCGA. We've run almost 4,000 samples across a number of diseases, and there's lots of interesting stuff coming out of that. But I want to show you what starts to happen when you aggregate data across disease, big data, and say, can it begin to inform us in a way that we don't usually think? Because I will tell you that the leukemia docs don't talk to the breast cancer docs, and the breast cancer docs don't interact with the ovarian cancer group very often, and it's the same thing on the research side because you're limited by the tissue. I'm going to start a little bit with breast cancer, and just as an introduction, transcriptional profiling, and Steve had a lot to do with this, has shown that breast cancer is many different diseases, 3 major ones: luminal, that we treat with hormonal manipulation; HER2 amplified, basal-like, we treat with chemotherapy; the HER2 amplified we treat with drugs targeting HER2. This has resulted in improved outcome. We've identified a new group that we call metaplastic or Claudin low that behaves totally different. This is improved outcomes, but it's resulting in problems. Breast cancer, the most common disease there is in women for cancer, is at least 11 clinically relevant different types that you should do trials on one group, not across. You will lose your data if you mix them up. They are different diseases, and I'll show you that in a moment. There's at least 100 new drugs about to enter trials, and we need to do combinations in the appropriate subtypes. The number of combinations exceed the total number of patients. So how are we going to do this better? We've converted breast cancer into an orphan disease, and so it will take a new approach. Now, this is the data. It's busy. I'm just going to give you the summary, but this is 11 diseases, unsupervised clustering, brought together so that we can look across and now have quantitative analysis and say that what we see in breast cancer relates to others. And the breast cancers sit together with the endometrial cancers and ovarian cancer. That's good. That's what we predicted. Colon and rectal is together. GBM and kidney cancer are separate. All of the lung and head and neck, smoking-related cancers cluster together. And then something went funny. Basal breast cancer and HER2 breast cancer has nothing to do with luminal breast cancer at all. As a matter of fact, it looks like a bladder cancer. And if any of you would have predicted that a bladder cancer and breast cancer look the same, you can have the Nobel Prize tomorrow. The other one that I love, the single best marker to tell you that you have a breast cancer, is the androgen receptor. Not the ER. It's expressed across a lot of these diseases. But if you happen to have the androgen receptor in this group, you are a breast cancer. And we can talk about why that is. But the thing is why is it that these breast cancers and bladder cancers come together? Can we learn something important by big data? And here is what it was. Here's the HER2 in breast cancer, and here's HER2. Actually, it's hidden underneath in bladder cancer. These are resistance markers, but it turns out the levels of HER2 in some bowel cancers and bladder cancers in this cluster are the same as those in HER2 amplified breast cancer. So why hadn't this been known? Well, we look at HER2, and we do most things by looking at DNA. This is protein. And if we look at the HER2 amplification, PIK3CA mutation, and the protein elevation, in these cases you see in bladder cancer it's about 7% amplified, but 20% have elevated protein. But if you look at breast cancer, it's only 11%, the HER2 positives that are positive. Endometrial cancer, serous version, the ugliest type, half of them have high protein, as high as breast cancer. That gives us a neat opportunity because there's a new drug out there that is dependent on protein expression, not addiction. And this is an antibody that binds to HER2, gets internalized, and releases a toxin into the cell. And the new studies coming out of breast cancer are incredible. And so we're rapidly moving down the pathway with this type of data to say, here's another group of patients who should be treated. We have a need, and we heard about this, is as we find out what's going on in patients, what do we do about it? And so we've built a big program to fill in that network questions from the IOM that Steve talked about. We have hundreds of aberrations in a single cancer cell. These effects are a combination of mutation, comutation, and lineage, as I talked about. Many of these are hotspots. I can figure out what a hotspot does by standard biochemical approaches. However, most tumors have previously uncharacterized aberrations in tumor genes, point mutations that have never been seen before. Do they mean anything, and what should I do about it? And then there's lots of genes that nobody's ever studied, where there's no data out there. So we want to know which of these aberrations are drivers, that is, if you targeted them, it would matter, and which are passengers. And what are the functional consequences of those events? And I'm going to introduce a lower organism genetic term here again. Hypermorph, increased activity but no change in activity. Hypomorph, decreased activity but no change in activity. And this nasty little beast here, where the mutation does something completely unpredicted. What we thought we knew is wrong. And unfortunately, this is what most of them are. And what are the therapeutic liabilities? How should they be targeted? And could we do this in real time for patients? Another approach to what Eric said. Does it matter? Now, this is a study from Boris Bastian looking at KIT. Several people in my team had shown that KIT was important in acral and mucosal melanomas. They did the initial work. Boris Bastian published the paper. The two young kids who did the initial work then had to come into my office, as they were both clinicians, and explain why this came from Memorial, which was unacceptable. But the data that came out of this is important. In a waterfall plot, there were patients that benefited and patients that did not. And what came clear and not surprising is that selection is important. If there was a recurrent mutation or the allele that had the mutation was amplified, you had a 40% chance of response. If it was a mutation that had never been seen before, melanoma is chock full of mutations, your benefit rate was 0. So even though it's a cancer gene and this gene is important in this disease, not all mutations are created the same. We need to know exactly what's going on for every single mutation. So take our complexity level up by several logs. Perhaps the best example of a neomorph is IDH1 and IDH2. If you go all the way back to high school and remember the Krebs cycle, you have IDH1 produces alpha-ketoglutarate from isocitrate. What's striking is the mutant produces 2-hydroxyglutarate from alpha-ketoglutarate, a new and unexpected function, not predicted. You could not predict it a priori. And these two molecules have markedly different function. If you block the normal function of IDH1 in an IDH2 mutant tumor, you are likely to make it worse because they have the opposite effects. This is a neomorph, and you need to know that. So, how often does this happen? I'm going to use endometrial cancer as an example for a little while. One of the things that I have to admit to, I'm actually a gynecologist by training, so these are all my areas, and I'm sure I'm the only gynecologist who would talk about systems biology or lead a department of systems biology, so it's there. But endometrium is a wild disease. It is ugly if it recurs. It's as bad as pancreatic cancer. Less than half the patients survive 1 year. It is the PI3 kinase-driven disease. It is a Cowden syndrome as part of it with PTEN loss. PTEN loss happens in normal glands, and they do respond to therapy. But I want to use this for another reason. There are 4 types of endometrial cancer. One is a serous that is mainly driven by copy number. Another that doesn't have a lot going on in terms of mutations. This is mutation frequency. We have the hypermutators, microsatellite instability. And when we started this, we thought this was the highest type, and it turns out there's another event where the DNA replication mediator, POLE, is mutated. These have up to 1,000 mutations per megabase. That is saturation mutagenesis, and it lets me do Drosophila work in humans. We study pathways in Drosophila by doing saturation mutagenesis and figuring out the effects. Well, nature did it for us. Neat thing is these patients do extremely well. And why they have this mutational load and do extremely well is something we need to figure out. But what a tool. We have a mutation state to explore. So we're taking data sets from our group, the TCGA, other places. Through a high throughput program, we can generate 500 mutant, sequence-confirmed, open reading frames a month and characterize them. My postdocs used to do 2 or 3 a year and we're happy. We're doing 200 to 300 at a time now. Now we can put those into sensor cell systems and say, do they improve viability, proliferation, and what do we do about it? And so we've put thousands of these into cell lines and know which ones increase viability and proliferation. Now this system only picks up viability and proliferation, does not pick up invasiveness, does not pick up metastasis, but frankly, as a physician, I don't care. I want to kill them. And if I want to kill a tumor cell, I want to know whether they increase viability or proliferation, and can I target that? Again, the team doing it. Whoops. So this is the P85 molecule that I told you I would talk about. There are a number of mutations in it. We express them in the system, and here are some of the ones that are positive. Background, wild type, and then the mutants all induce cell growth. Clearly, these are important variants. Some of them we knew about, things that were in the inter-SH2. This is the domain that binds P110, the catalytic subunit. But 2 of them were a little worrisome. That one was a stop codon, that one is a stop codon, and they're upstream. These molecules can't bind P110. They don't. They don't regulate P110. As a matter of fact, we've shown that this one, and published, regulates P10, the exact opposite side of the pathway. That makes sense, but I want to show you the story around this one, which is just unexpected completely. So the next step that we do is we take these cell lines that are interesting and screen across hundreds of drugs. It's an informer cell drug library that says, are these pathways involved? Would they make a difference if we targeted them? We start off with 145 drugs with a lot of redundancy, and this system that we use is nice. We addict the cell to our gene of interest. It normally grows with a growth factor called IL-3, and we can re-addict them to IL-3. So intrinsically, we have a toxicity assay, a secondary screen built in, and we can do thousands of these at a time. And what we look for is a difference with and without IL-3 driven by the gene of interest. So here's RAS, IGF-1, these 2 PI3 kinase molecules. Here's a series of different ways to inhibit the PI3 kinase pathway. The top PI3 kinase and IGF-1R, which is a PI3 kinase driver, uses this. All of them affect IGF-1R. KRAS, nothing here. Oh, an AKT inhibitor targets KRAS. That's now been confirmed, and we think we know the mechanism a little bit with mTOR. So here are 2 PI3 kinase mutations. You would think that if you had a patient with these, you'd target them with a PI3 kinase drug. Well, that's not going to do much good. No effect here. I told you it hit a different part of the pathway. This one's sensitive to this one. No effect here. It hits a different part of the pathway. This one's sensitive. Go down to the bottom. This one's sensitive. This one isn't. These are different, and we need to know that. But that's not the story. Because we looked at hundreds of drugs at a time, you could get an unbiased and unexpected view. Now, my postdoc was going to study the PI3 kinase pathway forever because that's what it had to work on. It doesn't. It works on the MAP kinase pathway. MEK inhibitors target KRAS and this mutant. JNK inhibitors target this mutant and not KRAS. The curves are the same. P38 targets KRAS and not this mutant. So we already have a story going on here that says we have a new problem that we have to deal with. So we then took our protein arrays and asked, what did these events do to signaling? And we could make some predictions. They should turn on those where they're sensitive. This mutant should turn on MEK and JNK and not P38. RAS should turn on MEK and P38, not JNK. Exactly what we find by looking at the arrays. So absolute concordance. We do hundreds of these at a time now. Go back to reductionist science. Here's the biochemistry. The Western blot shows exactly the predictions are right. The arrays were right. We need to do them because you can't publish otherwise. We worked out the pathway. We know that these things signal through MKKs. We could predict which one would be hit from what's known. Exactly as predicted is what came up. They hit different MKKs because they had different effects. We could then make the leap. And this is the one leap required by the postdoc. Why did this happen? Well, the MAP kinase pathway signals in the nucleus. P85 exists in the cytosol. Maybe this mutant was funny and went to the nucleus, and indeed, look at this. Wild type excluded, E160 excluded, this guy's in the nucleus. It brings the whole complex into the nucleus. It's all worked out. It's going to publication hopefully soon. But we could then go back to patients. And here are patients with that mutation. P-junk, P-ERK, exactly as predicted, massively activated and in the nucleus compared to a large number of controls. So we can go back and forth and have now a treatment approach for this patient that we hope would work. So the whole pathway has been worked out in exquisite detail, the mechanisms, the binding partners, and all of this. One regulates proliferation, the other anti-apoptosis. From there, it's obvious. Now, I want to switch over to what Steve talked about in terms of our major contribution, which is data generation. Now, I've shown you the data generation for the human cancer, the TCGA. We're building a human cancer proteomics atlas that others can work with and use. I want to show you two big projects. One of them is we've called the Kenna Proteome Project. We've knocked down each of the known kinases and kinase-related molecules and determined their effects on signaling networks using the protein arrays. Not one molecule at a time, but a lot of them. And we've pushed it a lot harder through miRNAs, because our hypothesis has been that miRNAs have evolved to regulate functions that will integrate at a protein level. It doesn't regulate molecules. That's what transcription factors do. This is a functional override, and it turns out that that is true. We're going to talk about rewiring of networks in both cases and the amount of data that we're putting out for others to figure out. We've also done chemical genomics, taking hundreds of cell lines, as Steve talked about, many different drugs, doing time-resolved functional proteomics analysis, and use this to build networks without prior knowledge. This is one of the key points here, is we are able to get around saying this is what we think and test it and prove it, but rather say let the data teach us with no prior knowledge, and that there is enough data now to do that. So here's the first project. It's a small one. We took these number of siRNAs. They were pools. We did them with and without EGFR. We did time resolved, 3 different time points. Basically, about 136,000 assay points in this particular experiment, about 80% of them that we went back and checked confirmed, and we found 2 important messages. First one is when you use siRNAs and look 48 hours later, you're not studying the effect of the siRNA. What you are studying is the rewired network that the surviving cells still have. If the siRNA targeted something that was important for survival, that cell is dead and gone, and you cannot study it. So it's not what you think. A lot of time was needed, but it's only the surviving cells. This is the networks. So we then went in, and I'm going to simplify this and look at one of the 42 candidates. And there are stories for every one of them. And this is phosphorylation of AKT with the different targets. This is a Z-score. And basically, there's a bunch that were just linear, did nothing, and then there's a tail here that altered AKT signaling. I want to show you one little story around that. And this is the network that was drawn, and this is actually a prior knowledge network. AKT is here. We're measuring phosphorylation of AKT and asking what each of the siRNAs did. We published this part of it showing that the MAP kinase pathway regulates AKT in a much greater manner than had predicted previously. Many of the predictions, or the things that were on here, were predicted from prior knowledge. These are all in the right direction, but there was a little surprise. This said that GSK3, that is downstream of AKT in every text, is actually upstream. That would be a real problem, and the answer is we went in and experimentally validated that. We knocked out GSK3. We had cells that didn't have it, and you can't activate AKT efficiently. The networks that we think we know are not right. And this is important to start from new data and a priori say, in the system you're looking at, how is the network wired? This is our miRNA screen. We looked at 179 proteins. It's about 1 million data points, 157,000 different comparisons to work with, and that's why we're loading it up for people to play with, because there's more data here than we could possibly look at. We've pushed this down to some data coming out, and I'll show you a little bit of it. Now this is our hairball. Everybody draws one if they're a systems biologist. It's required to get through your first course. And this is it. And what was intriguing is it's a major hairball of siRNAs by proteins. What was really surprising is we had one lonely protein out here that only one siRNA or microRNA regulated, and it only regulated one protein. That to us was a little surprising, and we've worked it out, and that's right. But this hairball is a challenge. How do you make sense of it? Well, we went back in and said, can we build a signaling network with no prior knowledge? So this is basically a Markov approach with Bayesian analysis. And what we found in doing this in 7 different cell lines is that these were found in all cell lines. This is not the way you would draw these networks today from prior knowledge. They're close, but there's things you wouldn't predict, like GSK3 beta and MAP kinase being key regulatory molecules of each other, and indeed they are. Others are things that we would have predicted. Few up here that we found only in the RAS line, so that says this happens in RAS but not in other lines, and indeed that is true. We've been able to validate that. So they are predictions that you have to go back and validate experimentally. We can also start doing this in single cell lines rather than groups, and in this cell line there's a very strong connection here between 2 master regulators that we don't see elsewhere, and indeed where RAS is mutant, this is very important. So you start to learn things that you could not if you started with prior knowledge. The whole key here is this is unbiased. One of the things we did is then ask which of the mirrors had major effects. And this is the unfortunate problem of science today. All of that data generation we didn't tell anybody about. We didn't tell the granting agencies. But once we had done it, we have to get money to pay off what we had already done. So we had to pick a hypothesis and pretend that we wanted to worry about a molecule and a hypothesis because that's the only thing that gets published and it's the only thing that you can fund. So we had to pick one. MIR124, completely unstudied. This turned out to be a very strong link in our networks. These are our master nodes, the same idea that influence many nodes around them as we just heard. And we went in and characterized them. And it turns out that MIR124 regulates a lot of proteins that are involved in cell cycle progression and viability, things we were interested in both negatively and positively. This was the most significant link in our different analyses by going across cell lines, so it was worthwhile deciding what to do next. This is what we saw when we looked at patient samples. This is across the TCGA set with 11 different diseases. This is the frequency of deletion. And so it turns out that in some of these diseases, ovarian cancer that I work on, almost 50% of them have deleted this microRNA. That's a pretty good hint that it's important. We actually look at levels, massive difference. Here's normal, here's tumor. We look at methylation, exactly as you would predict. And so here's our network with this as its master regulator, what goes up, what goes down, and what doesn't change. So now we could start to go in and ask, what does this mean? So we did the standard biology. It regulates integrins, P27 levels, motility, invasion, and proliferation, something that should be interesting. All the data is there. We then did a very nice experiment with Anil Sood and delivered this microRNA to tumor cells in vivo. It blocks tumor growth and induces apoptosis in vivo. So microRNAs are easy to deliver. They're stable. They're easy to work with. So here is the microRNA and the tumor weight. Add in etoposide, and there were lots of reasons why we would do that I didn't tell you from the networks. Similar stories to what you said. What did the network perturb? What would etoposide do? You put the two together, and it's a marked effect. It also blocks metastases. I showed you the motility experiment, and all of the predictions that we had in vitro of whether integrins would change, P27 would change, or apoptosis would change were confirmed when we went to the animals. So starting off with a big study down to low-hanging fruit and hopefully a new therapeutic approach for patients. So I want to thank the people that do the work. As I said, I talk about it, they do the work. This is the group that worked on the PI3 kinase network. They're a group that does our protein array technology. These groups worked on different stories. We do have a major Drosophila program in the lab that we do mutagenesis that we don't let us do in humans other than in endometrial cancer. This is our Kleberg Center and IPCT Center that works with the patient samples. And these are collaborators from around the world. To look at tens of thousands of samples, you need collaborators and people to help you with it, and a big bioinformatics group. Thank you.